We have with us Dr. Shiddamallaya N, who is a research officer of botany in uh, Central Council of Research in Ayurvedic Sciences, that is CCRAS, Department of Ayush, Government of India, New Delhi. He has completed his master's, then MPhil, PhD degree in botany from Gulbarga University, Gulbarga, and he is working. He worked for quite period in Gulbarga as a lecturer in different colleges, and he moved on to join this CCRAS as a research officer in botany in medicinal plant research, particularly section that is Central Council for Research in Ayurveda, Research in Ayurveda Sciences, New Delhi, Ministry of Health, Government of India. He presently he is assigned a nodal officer in major project research section in monitoring the medicinal plant research projects and activities in more than 12 peripheral institute of CCRAS involved in various disciplines like MEBS survey, pharmacognosy, then demonstrative gardens and cultivation, in vitro propagation techniques, microbiology under intramural research projects and collaborative projects. So he is a life member of Botany Department Alumni Association, Gulbarga University, Gulbarga. He served as research officer previously at CARI, Bengaluru, and RARI, Itanagar. He conducted various medico-ethnobotanical survey tours in forest areas of different states, namely Karnataka, Sikkim, Tripura, and collected plants, material, and herbarium specimens. He has organized various short-term training programs in survey of medicinal plants, identification of plant material, herbarium preparation, and museum management to researchers and academicians. He has successfully completed 8 intramural research, medicinal plant research projects. He is interested in interdisciplinary and collaborative research, developing new technologies of medicinal plant research suitable for research in botany. He has monitored researchers of PhD degree in botany, Mysore University, Karnataka, and I think successfully guided three PhD candidates to their PhDs. Dr. Sidmalaya has published more than 70 research articles and 8 books, book chapters, and monographs. He is a member of editorial board of reviewers of some index journals, particularly published under by this Ministry of Ayush, particularly. So they publish almost 4 to 5 journals. So he is one of the editorial members and reviewers. So with this brief introduction, I welcome Dr. Shiddamallaya N to this 16th refresher course in life sciences. So on behalf of all the participants and on behalf of University of Mysore, then HRDC, UGC-HRDC Center Mysore, as well as Department of Botany, and on my personal behalf to this refresher course, sir. And I request Dr. Shiddamallaya to present his talk on future prospects of medicinal plant research. Over to you, Dr. Shiddamallaya, sir.
Thank you, Rajkumar. Am I audible to you all?
Yes, sir. Audible.
Yes, sir. Yes, sir.
My
Audible.
slides. My slides are seen?
Can you please?
Yes, sir.
Yes, yes. Yes.
Okay.
Can you go for a slideshow mode, sir? Please, sir.
1 minute. 1 minute. Okay.
Yes, sir.
Yes, sir.
Fine, sir.
Yes, sir.
Thank you for the invitation, for taking part in your 16th refresher course in life sciences online mode. Really, I'm feeling very happy. Somehow I am joining my Karnataka team. I'm I think near about 7 years away from the Karnataka. I think after a long gap, I'm getting my friends in touch. So with this, I will start my presentation. Actually, what I thought regarding this, my presentation means, when I discussed with my friend Rajkumar, he said about that bioresources for sustainable development in the modern era. And when we discussed about the topic, he said about I have to speak about the medicinal plant research, that too with reference to the future prospectus. 1 minute. Excuse me. Hello. Okay. Yes. I thought here we need some integrative approach in that. Actually, what is happening in this academic line, we will do some part wise. I can give so many examples. It may be tissue culture, it may be pathology, it may be any other taxonomical line or any other identific. This one we can say systematic botany, embryology, like that. A piece it is going on. But I thought here I have to give us some integrative approach. A complete approach should be there. Whatever we do in that medicinal plant, I thought to choose my own department work to present here how we are working. That may give a clear-cut idea to all how we have to work. Individual work will not give what we expect that much credit. When we work in a team, that will give a good credit to us, and that work it may have a good impact on the society, like that. So for that I choose this topic. So we know that medicinal plants are very important, especially in that health related matter, because of that medicinal plant, when we are saying, means those plants are specially used for the therapeutic purpose only, that treatment purpose only. But even in the modern medicines also, we are getting some of the herbal products. But the situation is something different in this allopathy. What is happening, that herbal drug, how it is entering means, we are identifying one of the biocompounds or a biomolecule, then we are going for the in a synthetic line, or sometime we may using the same thing, we are entering like that. But actual our Indian system of medicine is in a different ways. In the Indian system of medicine, other systems also there. It may be Ayurveda, it may be Siddha, or it may be Unani, or it may be Yoga, Naturopathy, Homeopathy, and one more recently added as a Tibetan medicine also. Here we think that whatever the plant part we are using, that is a complete part itself we considered as a medicine. That formulation, what the medicine we are saying, that may be prepared from one particular raw material, it may be more than one, that one we may say that compound formulation, where we are using some different plant parts together, or sometimes we are adding some other minerals also in that. Some of the medicines are there in this Indian medicine system, some animal products also used. But whatever it may be, it is a whole thing. But in the allopathy, it is not working. It is a little bit different compared to this. They are concentrating on the molecules, we are concentrating on the complete thing. But in this, I want to tell how we start our work. We have a lot of literature with us. So many Vaidyas are there, our saints are there, their written scripts are there, manuscripts are there. With that, we are following. Apart from that, so many other information is lying. That too, especially in the tribal areas of India, and some age-old peoples in the rural areas also having a lot of information. But we are not documenting. Other people in the academic line, we are thinking in a different way. But CCRS, especially Central Council for Research in Ayurvedic Sciences, we are thinking in a different way. We wanted to document that first. We want to enlist that, because that is one of the good piece of work. We have to do that first. Then we have to continue with that. So that's why first our work start from the literature. First we will see that what are the areas are there. Suppose if you wanted to work on any particular state or any forest, we will study first literature. Are there any tribal areas? Are there any traditional healers? Are there any published literature available with that? Then we will see that are any particular disease they are concentrating. Like that, we will collect the data. Then we will document that information, whatever it may be, that folk medicines. Then we will start our work. It means we have to start from the base, and that has to reach to the end of the, that end means that preparation of medicine, and then that reach to the patient. In that way we work. It is a teamwork. Actually, CCRS is having the 30 institutes in the India. Every state is having minimum one institute. In that different works are going on. It may be botany, it may be chemistry, what you are saying, pharmacology is there, literary research is there, clinical trials are there, many things are there, but all together we work here. We are not working individually. It is a teamwork we have to do. I expect the same thing even in the academic line also. When we give a topic, a PhD topic to the student, that is a piece of work. But that continuation work we are not getting. Because when we screen for the papers or when we screen for the PDF fellowships in our institute council, that time we face these problems. So that's why I thought I have to give the complete picture of this style of work required in this medicinal plants. Like that, these are the different types of medicines followed in this Indian system of medicine. In this Tibetan medicine is not there. So I can give one more clear picture is this. Here we always think we wanted to live healthy, we wanted to live happily. But when we think about the first, we have to, I'm discussing about the allopathic system of medicines where surgery is there. Even that is also there even in the Indian system of medicine, and special treatment techniques are there, different styles of techniques they are using, and supporting measures are there. Suppose any patient is admitted, some supporting things also they need, that is there. Diagnostic techniques are there, different instruments they are using, technology they are using, then they are giving the medicines. But what we require, healthy lifestyle is a very important. This is one of the separate part. When we think about the traditional system of medicine, here is the preventive measures. We have to prevent the disease that should not come. But that part we are not finding in this allopathy. How to maintain our health, we will give. Sometime we may tell that what to eat. In a season wise, we will give what are the things we have to follow in our life. So all these will comes under this preventive measures, but that is not there in this. And curative medicines are there. Medicines are very common. Here we are giving the medicines, but most of the medicines in the allopathy is symptomatic one. But in this Indian system of medicine or a traditional system, it is not like that. It is proven from thousands of years. Now we are working on a symptomatic. Suppose we are having the headache, we are taking the paracetamol, but we don't know that headache is for the acidity or what are the for other reason, or that may be migraine. We are not thinking that headache as some common medicine we prefer beginning, but that should not there. That is not there in this traditional system of medicine. And some alternative measures are there. Even in the traditional system of medicine, surgery is there. Before allopathy, some surgery practices are there in the traditional system. Some records are available on that. Now that for research areas, how we will work. First one is a literary and fundamental research. What the available literature is there, that one we have to screen it. Suppose we wanted to do any work. Even the same thing happens in our PhD work also. First we will do survey that literature available on our topic, literature survey, what we will do like that. Here the classical literature survey we will do. Then that fundamental research. Fundamental means a basic thing. What the basic thing we can do? What we study in our BSc, that is a basic we may say. In a PG level, a little bit high. In the PhD, special a particular on top one particular topic we will work that like that. Here also in the Ayurveda also some fundamental research are there. Some principles are there. For example, I can say that we can say Rutucharya, a particular season, what type of cloth we have to wear it, what type of food we have to take it, some basic things. Suppose we are feeling little bit cold and cough, what we have to do, some basic things like that. Then medicinal plant research is there. In the medicinal plant research, this work start from the field collection, processing, then pharmacognosy, phytochemistry, then microbial load, heavy metals, pesticides, all we will study. Then formulations, preparation of formulations. Then once the formulation prepared means pharmacological study, safety and efficacy study will take place in there. Then next we will go for the clinical trials. That is the thing. Medicinal plant research, drug standardization, quality control, we have to maintain it. Suppose what happened, quality control is very, very important in the raw materials. We don't know that we collect the raw material from the different parts, but that all the plant materials may not be same in their phytoconstituents. We have to screen it. which one is matching our standards. We have some pharmacopoeial standards, which are the material matching with the pharmacopoeial standards. That one only used for the preparation of formulations, like that it goes. Once formulations prepared, next we will go for the pharmacological research. Then it will be clinical research. One more step is there. I will tell you later. That will come next. Then that same thing I am telling that R&D, what is that, the documentation and validation of a classic Ayurvedic formulations and generating evidence on safety and efficacy. In which areas we are working? Some major areas I am telling. Already classical medicines are available. We know that this medicine is for this, but present condition it is different. We are following the Western systems. We are preparing the document according to the Western system only. We need that all the thing is, it is evidence based. We don't have a evidence for that. We are using so many things. We are taking the medicines. Even we can say that what the spice box is there in our kitchen, that is a first aid box as per the Indian system of medicine. Suppose cold cough we feel, means we use a pepper, we use a ginger and so many things, but evidence we have not developed. So that evidence development is a major work of is going on in our CCRS, and this is required also. Sometimes we are taking the support of other institutes, other research institutes, other universities. We are taking some of the good PG centers wherever that lab facilities are there. But we are thinking only that government institutes only we are concentrating more. So if anyone is having that good instrument facility, they can work in collaborative way with us. This option is open for the all the researchers, especially in this Ayurveda aspect. And the development of safe, effective formulation, that is very, very important. Whatever the medicines we are giving, at the time of preparation, we have to take utmost care. And when the collection of raw drug, I can give one example. Anacardium accidentale, that seed is one of the important thing, important raw drug for us. But we cannot use the seed as it is. We have to remove some of the unwanted things from that. We have to purify that, then we have to use it. And then we have to think about the Caesalpinia bandu. There also some of the other things are there, toxic things. So that one we have to remove it, and then that one has used as a drug for the preparation of medicine, like that. Where we have to see that the safety, what is the effectiveness, how many days it require, how much quantity we have to give. All this we have to see that. And same time that management of disease, national and global importance. And here disease also we select. Every year 3-4 diseases will be selected. What we will do, 4 diseases selected means then we will screen which are the medicines available in the markets. Then are any already prescribed medicines are there by the any doctors. That data also we will collect, and then we will check the literatures. Is there any preferred formulations? Then that one we will study it, and that one will be introduced to the society, like that. And capacity building in core research areas. And side by side we will develop our labs, and we will develop collaboration with a good lab facility persons, and we will provide the training, like that. We are doing that capacity building and translation. This area I left, I have not discussed earlier, I am discussing. See, whatever the medicine proved clinically, that one we are giving to the pharmaceutical industries. Suppose it is already mentioned in the text, simply we are preparing the data, and that the data we are providing, that data will help them, the industry people, to market it, that product. It may be nationally or internationally. That is a one part, that is a industrial support. And another one is that suppose we are collecting any information from the traditional healers, suppose it is proved something good, that one we will give it to the industry. One NRDC is there at the middle, that NRDC will help us to give that data to the industry, or some MOU may be there, MOU will be there. And what happen, we will get some royalty on that. In that royalty also, some part will be remain with the NRDC, and whatever the part it will come to us, in that 5% we have to give that National Biodiversity Board, and in the remaining amount 50-50 we have to share. Who has given the information to us? That healer. And who has given the data to us, he has to get the benefit. That is our main motto of the CCRS. And that remaining 50%, why we take means our expenditure is there to for a single formulation, minimum we need 10 crore rupees. That much we are investing in the different steps or a different level. So that one we wanted to collect it back, but I think sometime we may not get that much, but whatever it may be, that maybe government money we are adding, something we have to give it to the government, like that 50% we are keeping, 50% we are giving to the person who has given the knowledge to us. And some publications also we will do. Hundreds of publications are there in the medicinal plant. Near about 40 to 45 publications are available from the CCRS. Some are free are there. Some are freely distributing in the schools and colleges, some universities sometime, or some are there online. And we have our own journals, 3 journals we are having, JDRAS, JRAS, and one more that manuscript purpose, one more journal is there from Hyderabad Institute. All in these 2 journals are UGC listed one. This is a free for publication. Here we are not charging because this is a government institute, free we are doing that. Then the dissemination of the research outcomes, whatever we do, that one we have to disseminate it. Sometimes we are developing the agro techniques. We have our own tissue culture labs, gardens. We have conservation of species is there. Whatever the data we are having, the data we are giving to the researchers, and we are taking care that should reach to the common public, their benefit. Some brief, I can say that only related to medicinal plant only. We have some 12 survey centers in India. We have covered 947 surveys we have conducted, and 964 forest divisions we surveyed. That much data we are having. That data will help us to identify that particular species, where it is available, what is the quality of that material. We can tell that because the pharmacognosy also we are doing, and that phytochemistry we are doing. That because whenever we prepare any formulation, we will do that work in triplicates. 3 geographical regions samples we collect. Then when we do that any phytochemistry, that again that each sample in triplicates, then that on and average data will be used for the preparation of the monographs, like that it goes. And even the pharmacognosy also, more than 500 plants we worked that. 7 laboratories are there in India. 3 volumes of books we published, that is given. This is available in all the Ayurveda colleges, and some of the universities also they are having. And if anyone is in need of the books, means they can get it from the council, that is available. And some of the agro techniques also developed. But in the agro techniques, something I'm not getting a good thing, because here it is going piecemeal. 4 or 5 plants we are having the complete thing. Some tissue culture part someone is working, or that our intention is that whenever any new agro technique developed, means that last end, that plant part we have to collect it. And same time we have to collect the same material from the natural source and from this experimentally grown one, and then we wanted to compare it both. When it is giving the good result, then only we can say that is a good agro technique, otherwise that is a waste. So like that, it is work is going on. And even some in vitro propagation work is also going on. 14 species worked in that. I think I can say that 70 to 75% forest areas data we are having. A lot of publications also there, but exploration work we will do that. Medico-ethnobotanical survey we conduct, and we will enlist the plants available in that. Complete list we prepare. Then we will bifurcate which are the Ayurveda, which are the plants used in Ayurveda, which are the plants RET species are there, where we have to put a more concentration on that for the conservation. Like that, work will goes. And next is the local health practices. This is a very important. This is a base for our work. That too, especially for the tribal people and this old populations, village people. From that we are collecting, and collection of genuine raw material. And nowadays I am noticing that some of the taxonomists, I think in India, we came to know that hardly 250 well we can say that well-known people or well-known taxonomists are available. Only 250 people are there. Some people are what we can say that's doing some business in that. They are giving the service for the identification of plants. They are charging. But Botanical Survey of India is there and our CCRS is there. We are identifying, but some it is a limited chargeable way. Some what we can say consultancy they are running. But this consultancy business already there in the foreign countries. When I am studying in the MSc and the PhD, I came to know that identification of algae, there is a consultancy in the foreign countries. We have to give the material and they will identify and they will give like that, which are not identified in India like that. And I met some of the peoples like that also. So some consultancy is going in India also, especially in the angiospermic plants. These people we are noticing mostly in the Ayurveda colleges, Dravyaguna persons, their knowledge is limited to that Ayurvedic medicinal plants. But basically here a botanist who are there, who are studying the taxonomy in the botany, these people are having the good knowledge. We can identify the many plants compared to them because their knowledge is a very limited, their formulations are so limited like that they are doing. So that is one of the good businesses going on, or some people are maintaining the plants and they are giving the raw material, they are selling that for the research, they are charging very heavily. This is a type of businesses going on. But sometimes I feel our botanists are turning the entrepreneurs. This is a good thing. They have to do that. Simply we are studying and we are born to work under somebody. That's not good. As a botanist, we can do many things. Own business. Always, whenever I meet with the students, I will tell the same thing. Don't be a staff to work under anybody. Better to develop your own thing. You can do that. So many opportunities are there with us in the botany subject, but we have to turn that. Then only we can. Only botanist can give the genuine material. No any other persons, because I can give one example of in the Indian system of medicines who are studying this Dravyaguna or taxonomy part, they have their own terminology for that. I can say one plant named Bala. Here we can say Sida cardifolia is the exact genuine plant material. But when we say the Bala means all the Sida species they consider as a Bala. So sometimes they may not in a position to identify the which is the actual or a genuine plant in that. So here botanist role is very, very important here. So that one can be encashed by all the botany persons if they wish. And passport data preparation is also very important. Here collection methodology is there. I will discuss later how to collect, what to collect, where to collect, when to collect. That is a separate thing is there. Whenever we collect the material, along with that we have a passport data. Minimum data we have, in which area we collected, on what date we collected, who has collected, and that plant description little bit, and that plant digital photograph we keep. Because we have to know that that is a tree or a herb or a shrub, whatever it may be. Simply we are taking the branch photograph, we may say it is a shrub or a tree, it is very difficult. Different pictures we are keeping with in that digital passport data. And GPS reading also we are tagging with that. That is very, very important. Suppose we need that same material, means we can go and visit that same spot and collect that thing. And then that same data will be published in the form of research papers. Here also some restrictions are there. I will tell you that LST how, which are the information we block and which are the information we can publish. I can say that 12 our institutes are working in this line. Here what happened in this medical ethnobotanical survey, here only maximum concentration is there on that medicinal plants used in Ayurveda, and along with that others also we are taking care. And I wanted to give us a small picture of that, how we will do that. Here I can say we have some different gardens are there. If anyone wanted to visit that garden, he can visit it, or any particular plant he wanted to see personally or physically, he can visit and see that. And some agrotechnics successful technologies are available in the CCRs that can also they can use. And now I can concentrate on this selection of plant species, especially for the medical ethnobotanical survey. Suppose for our research, it may be PhD or any other or anything, whatever it may be, anything. Suppose any herbaceous species are there, means a particular season we have to concentrate it. So for that we have to refer our flora available, and same time any published literature available. But nowadays one more problem is going on. So what the floras are there, old one, these are not matching with the seasons. That flowering and fruiting, their season is changed. Because of this global warming and other things are there, some other issues are there, this other factors are influencing on that. It is not matching exactly, but 75% somehow it is matching with one another. But we have to see that what the species we have selected, that is a herb, means that particular season we have to select it and do it. And trees and shrubs, we have to see that where it is available, where we have to refer the floras, and they in that sometimes we have to see that the habitat also, what type of habitat. We can get it on the hilltop or a slope, or it is a terrestrial or aquatic, it is a parasite or epiphytic. We have to know all those things. Then only we can collect the genuine material. Collection trips also, I wanted to say something. We do that like that. Suppose one day trip we can plan for the raw drug collection, or suppose any species is there that is very problematic. We are facing the problem of identification, where we will plan it. In that forest area, we will survey the complete forest areas. Are there only one plant, or we can get many number of plants of same species like that, or in a different states it is available, or a different districts available. We have to collect the data and we have to plan for the exploration and even expedition also. This is a years together work we can do in that. Such type of work we can give it to the PhD. I wanted to give one more small information to you all. We have 8 international recognized herbarium. Any botanist may need help for the identification of plants. They can take the help from these centers, where you will get an accession number, but you have to deposit herbarium there. Because now that especially for the publication of flora work, where we need this accession number is compulsory. Or if you're going for any phytochemistry work you wanted to publish in a well-reputed international journal, they will say who has identified, any botanist name we have to give that, who has identified from the which institute. Is there any accession number? Without accession number, it is not accepted. Even in clinical work, what I am saying, that formulation, preparation and other things, there also we need all this data. Otherwise that formulation may not be approved for the further for the marketing and other things. So that's why now we have this 8.1, we can use it for yourself. Now I am telling about that some brief of this LST data collection and what we have to do. We have our own format, local health traditional collection format. This is approved by the Ministry of Ayush. This is well practiced even in the CSIR labs and other DST research centers. This same format they are using, because this is having the complete data of that. But what we will do, some mistake we will do as a botanist, when I am in a PhD, I came to know that so many problems are there. A botanist will meet the traditional healers, collect the data. But that data we feel authentic, but that is not authentic. There is a problem, especially for the identification of the disease. We may, as a botanist, we may not in a position to judge that what the disease he is telling. Is it right or wrong? I am also having the experience of the 17 year. I worked in the field. I have also collected the data. I interacted with the hundreds of traditional healers. There I faced this real biggest challenge. So what we will do, we will keep always, we have one of Ayurveda physician as a member, as a team member with us whenever we collect the data. I prefer who are doing the work on the LSTs, we have to keep one doctor with us at the time of collection of data. Especially for the identification of disease is very important. Plant identification we will do, but the disease identification, sometime it may go wrong, means everything will go wrong. That's a waste. So that's why always one doctor should be there whenever we document the traditional practices. That data, it is having a minimum about 40 to 50 columns that data will be filled, and that claims we will bring to our institute. Then that data we will match with the text available in our libraries. Otherwise, we can say that that will be matched with the classical texts. It may be Ayurveda, it may be Siddha, it may be Unani, whatever it may be. We will match that if it is matching with that or not, or it is a unique, where we will segregate that LST claims. Suppose it is matching with the data, that one we will publish it easily in the papers. If it is not matching, means that is a very, very unique claim and important claim. Then that same data again we match with that any published literature it is available in a journal.
So it's not.
Hello, sir. Sir, your voice is not audible.
Hello, sir.
Is it audible, madam, now?
Audible.
Yes, sir.
Okay.
Yes, sir.
This consent form, prior information consent form is very, very important. We are not taking the consent from the knowledge provider. If we publish any research paper, if he put any case on us, means it is very difficult to come out of that. We have to get a primary consent or a prior consent from that healer. You have to prepare your own format. It is a, suppose if you refer that TKDL format, means that is about near about 15 to 16 page data is there. So if you read that, you may not in a position to do any work on that, because so many clauses are there in that, legal clauses are there. So make a brief data, this format from your side, and get the consent. Simple format we say, if it is matched, we will go for the publication, or if it is find unique, that will be not disclosed, and further study anything is there, means that will be carried out, and whatever the benefit we get, that will be shared. Like that simple format we have to prepare and get the signature. These are the steps, the same thing what I said, that how we will screen the data, that validation of holistic claims, that 4 claims we have to do. And if that data is matching, means we are publishing books on that. If any novel, that novel claim get, means suppose if you get 1 or 2 novel claims in a year, means that is a good contribution from our side. In that way we have to work it. So we have collected a lot of information, and already so many books are there. In that 2 important books we have published. This is available for the sale and other things. And some of the claims I said that here, these are the leads from LST. These are the products prepared by the CCRS and given to the industries. And what are the benefit we are getting? That benefit also shared to the concerned people. These other things are there. Ayush 64. Actually, this Ayush 64 medicine that came out during the corona, that has given a very good result. Here we have not noticed any death who has taken that Ayush 64. That much a good medicine is prepared. But earlier, that was the medicine preferred for the malaria. But when we screened that all the information, that time we came to know it may help in that corona. We tried, but that has given a good result. We have noticed some of the patients went to till ICU, but we have not noticed any death who has followed that use of this I-64. Like that, so many products are prepared and given to the industries. It is a routine procedure of us. How we will develop this thing? One more, our wing is there. We will work in that way. Identification of priority area, a particular disease. Sometimes we concentrate. Now we can say diabetes is a major thing, cancer is a major one, and obesity is there, tuberculosis is there. These are the diseases selected. Now we are working on that. We are doing the literature service. Some hypothesis basis. How can it may be cured, or any information is already available, or what the medicines we are using, are they giving any good result, or any other medicines using for some other disease that may have any good formulation, what we say, that good molecule in that may help us, like that we will say. Or first we will say any intervention. Suppose I can give that one example, cancer, that chemotherapy what we are taking for the cancer. So where we will give some interventions at the middle that will not affect on the health of that patient. Some interventions first we will give, or sometimes we will replace the complete allopathic medicines. In different way it goes. Then standardization, preclinical safety study we have to do that as per the applicability. Biological activity we have to check it. Protocols we have to prepare. This complete CRF should be there. Clinical research profile file we have to prepare it, and that has to be registered. Regulatory requirements we have to see that if we have any formulations we cannot give to the patients. So some regulatory aspects also there. Clinical trials we have to conduct it. That to first OPD level we will see. Then we have need more patients. We will adopt some of the institutes, colleges, this medical colleges. There we will do. Then we have to see that IPR values any as applicability. As per the applicability, we have to share that profit and other thing. Sometime we may go for a patent, or sometime we will register our technology, like that, and then commercialization. Once it is commercialized, means that benefit sharing is the next one. These are the some of the information I wanted to give here. What are the minimum things? As I think in this discussion, most of the botanists may be there. I feel like that. So that's why whenever you plan for any work to enter in that forest department, that forest areas, see some of the things we have to keep in mind. So that's why I'm discussing. Usually what happens, we will do any study tour means directly we will enter in the forest, or roadside we will collect. That should not be there in this research. Minimum 1 kilometer distance we have to maintain from the road, we will tell, because of that pollutants may enter in that plant body. So that's why we have to concentrate on that. If any plant we want to collect, means first you should know about the laws of the forest departments, this wildlife, or it may be biodiversity board or conservation acts. All we should know, because by mistakenly if you remove one rare, endangered or threatened species, means it is very difficult to replace that. So that all the knowledge should be there. What part we have to collect, how to collect, all the should knowledge should be there. And botanical identity, we should be thorough in identification of the plant. Then only we have to collect it. Otherwise, better not to touch the plant. We will say directly. We will say so many times for the students, like that. So it is a must. Otherwise, that is a waste of the plant. And appropriate developmental stage. We need flower means flower only we have to collect it. We need a leaf means matured leaf only we have to collect it. And sometime in the fruit, immature fruit we need, or a mature we need. What we need, that one only we have to collect it. We should not touch the other part. We should not disturb the other plant. We don't have any right to kill any other living things. That plant is also one of the living organism. We don't have a right to kill them. And that plant health has to be seen first there. Suppose that the plant is infected or anything it is happening, or it is not showing the good growth, stunted growth is there, that plant material should not be collected for the research purpose. Otherwise, what happen? It may have added some other compounds in that extra thing for the defense purpose. It adds some other molecule in the body. Suppose we are concentrating on a particular molecule, we can. Otherwise, for the general study, that may not be collected. And after collection, we have to that dry conditions we have to see where we have to dry, how it is that the soil, what is the rain, what is the humidity in that area. That data also we need. That data help in a different way. I will discuss once again that in the later. That is very important thing. This collection. This can add many things in the research. I will tell you later. And equipments. What are the equipments we are using? And here some of the equipments are not used for some particular plants. Where only wooden things only we have to use. Iron things are banned in that particular plant. Whenever any latex is there means that where we are iron thing is there means come in contact with that means it may react with that. So many things are there. So that things we have to follow it properly. Now, who is working, who wanted to work on that cultivation line? Where what the things we have to concentrate first? Our main motto is their mass production. We need a maximum raw material. Whenever we are planning for the agro techniques, or whenever we are thinking for the tissue culture work, definitely we select rare, endangered, or threatened species, or economic important medicinal plant only we select. But our main motto is that mass production should be there. Maximum growth should be there. Like that, we have to plan it, whatever the plant we are selecting. Which are the plant available luxuriantly in the field. It is not necessary to go for the study on that. Which is not available and which is economically important, that plants mass production is very important and fasten the growth. I think some of the plants may need 6 years or 7 years to reach a particular stage, and that stage we are collecting the fruit and seeds. So is there any technology or anything we can do through research to reduce that growth period, or we can increase the yield, or it may help to conserve the RET species. We can increase the number of RET species. And nowadays, last year I have started something different. We know that naturally grown plants are having the good source of phytochemicals in that. But last year I thought, is the hydroponic system may help us or not? We started the work, but initially it is giving the good result. Growth, it is good, but end result we are waiting. One more year I have to wait. That I wanted to compare that hydroponically grown plant with the natural one, and where it has to show the similarity in the phytochemicals. Then only this plant material I can use for the further thing. So such type of work it is required. And some of the plants we need that resistant to the some of the microorganisms or fungus and other things. Or nowadays some nanoparticles work is also going on, but here in the Ayurveda it is a limited. I think after another 6 months or 1 year, some Drug and Cosmetic Act rule is changing. If that changes means whatever the work that anybody doing on the plant research, that will comes under the Indian system of medicine. But what I can say that herbal industry, what the synthetic things they are using, they are not with the Ayush, they are not with the health department like that. They are at the middle. No one is supporting, no one is handholding. But now it may come to the Ministry of Ayush. Once it comes means they will get a good benefit and good research can be done in that. Some of the references, some of the ideas I wanted to give that this one can be tested. What I said that basic Ayurveda, what I am saying that. So some basic concept are there. Are they really working in the present condition? We have to check that. That is also one of our responsibility. We have to do that. For example, this Madanaphala Cottonaregum spinosa. We know that that is known for emetic, and some of the people are using to control that alcohol habit. Who is taking more alcohol means they are using that. Just it is causing the vomit like that. And but usually that one we collect in the month of, especially we can say Grishma Rutu, summer season. That one we have to check it. Is it really works that seasonally collected seed or not? That one we can see that. Like that Amlek is there. That best time to collect the fruit in the Pushya Maga Falguna Mas, what we can say January to March. But the thing is that nowadays this Amlaki fruits, this one Emblica officinalis are available throughout the length of the year, but we don't know which one is correct or when it is collected, because when we go to the market we will get a dry sample, a dry material. We don't know exactly at what season they have collected. So we have to do the study on this seasonal variation. This is one of the part of our work. We are working on this. I think near about we worked on 56 plants till now. Which season is suitable for the collection of raw material, at what stage we have to collect, and that can be used for the preparation of formulations like that. Lagenaria cinerea, so that suppose we need that tender leaves, so what is the season we have to see that. So some of the examples I have given. So like that you can plan your research work on that. You can publish a good research paper. This is one of the good contribution to the society, because so many people will follow that. And some of the already available literature is also there. So some are showing a little bit controversy, some are matching. Suppose it is matching means if it is true or not, we have to check it. Suppose it's a controversy means we have to see that which one is correct in that. Like that we have to carry out the work. Sometimes what we will do when we check in the literature, if it is someone worked means we will stop that work. We should not. We have to see in different things. Here in the research, negative also a good contribution. Suppose we are getting the negative result means we feel unhappy. That is not like that. That negative result also sometimes it will give a good credit. So like that, in that way we have to think it. That collection procedure we have to just I have shown that how we will collect like this. Suppose that latex we are collecting means in the plastic things we are collecting, we are not touching directly. Suppose root we need means a part of the root we are collecting like that. Suppose we need a stem bark means a piece of the bark we will collect it like that. That has to be carried out. This one I wanted to discuss with you that regarding the specially collection. See, this can add a good piece of work in your research. But what we will do, a single person we will collect from the single area and we will do that phytochemistry and we will say this is my sample is very good. Not like that. Or sometimes we will say, suppose when you collect that material, we will say, suppose you are getting some negative aspect means that plant material is having the negative aspect. But you are not mentioning the what from which areas you have collected. That area material is having the some negative impact or a positive impact. But simply we will say that plant material itself it is a wrong. That should not be. Here we have to collect from the different geographical conditions. Then you have to go for a comparison study. Then you can say that which one is good, which one is not. And same time it will help you to identify the which area is good for a cultivation. You can help the farmers that particular area is suitable for the cultivation of that geographical area where that plant can be grown and sold to the industries. Even industry also searching such a type of information, such type of information you can provide, but it is a teamwork. One has to help other, because it is a very easy to do that. When we do the MSc, when we do the PhD, we have a lot of friends. When we have any interaction with the conference and other things, we have a lot of friends. So with the friends, you have to collect from the different geographical area. You do some comparative study, come to a conclusion. That is a good piece of work, where you can say whatever the positive result or negative result, that is good. But simply working on a particular area from the particular geographical region, you collected the material, you are assuming that is a complete. That no, that should not be. Especially for the cultivation, here some of the factors also we have to concentrate. What I said that earlier study purpose only, these are very, very important. We have to see all those things, especially that physical factors, climatic factors we have to see, that soil we have to see, that relief, that what type of terrain is there, that is what we can say, the topography of that area we have to see, and what the institutional factors, what the institute is involved in that. Sometime what happen that land will be on lease, or land reforms has been done or not, from which areas we have collected. Same crop we are repeatedly cultivating means where we may not get a good yield from that. Like that, even in the medicinal plant cultivation also happens. That one we have to see. Infrastructure we have to see. So many things are there. Now that the cultivation is one of the business, medicinal plant cultivation, that can be. Then some of the guidelines I wanted to tell, I think I reached 3. Time is very limited. Some API and WHO guidelines are there, classical texts are there, and guidelines for good storage practices, it is available. How to store the raw material, how to collect the raw material, guidelines are there. How to store the raw materials are there. For all the things, some guidelines are there that has to be followed, that has to be recorded in your publications. Then only that is accepted for the patenting and other thing, or for the further work. This is the storage of drugs, how to pack it, what temperature we have to maintain it, what are the other precautions we have to take there. This is about our museum, but industrial storage is a completely different, because I know that even in your colleges or universities labs also you are preserving. Earlier we used to tell Ayurveda is related to only medicine, it is concentrating on the formulations and treatment, but it is not like that. It is concentrating on all the other things. Now we are working with the IITs, we are working with the good laboratories, and we are quality controlling, tissue culture work we are doing, so many things we are doing, even biotechnological things also we wanted to include in that. But we want to get a good product from the plant, a good raw material, like that. So you can think on this. And especially microorganisms, microbiology is also one of our part, where we are concentrating on that identification of a particular microorganism which is destroying our raw material. Here we are not concentrating on other thing, but in universities it is a different. If you work on any microorganisms, means next you can think about that enzymes and other things, what that phytochemical you can collect it. That is a different way, but here it is a different. In the Ayurveda we are thinking in a different way. What are the disease causing, or what are the raw material destroying things are there? That one we are concentrating. And in which area that is microorganism is harming more, in which area it is not, what are the factors are affecting on that to control that, or which are the factors are supporting that for the growth of that microorganisms, like that we have to work it. One more area is there now, that alternatives. Here what is happening now, we are not getting the sufficient raw materials, so that time we are thinking for the substitute and alternative for that. So for that different strategies also developed to identify that. It may be phytochemically look similar, and it show good that when we do that preclinical studies where it has to show their activities in the particular line, in that way we are identifying the other things. This is for the just production of that plant, what we need in maximum quantity, which are the techniques we are following. In vitro is there, hydroponics is there, hardening of plants. Well, you will do commonly in the universities and in the labs, you will do like that only we are doing. And this is a drug standardization, how we will do here. But first our main motto is that quality assurance is a very, very important one. That begins from that quality control, we have to check it. It may be quality control of raw material, and it also quality control of prepared medicines. Both the places we will see that the quality control. And some reference standards are there, with that we will compare with that. And pharmacopoeia standards we follow. And suppose if you identify any new plant, it is not having not listed in this Indian system of medicines, you can add but having that all the data with that. I can tell one thing in that Indian system of medicine, simply we can say 600 plants are there in Ayurveda, but in that hardly 50% of plants are having the complete data. Remaining plants are there, they need to work. So you have to identify, you have to see that. I can say that pharmacognosy, phytochemistry, these things anyone can be done, and some organoleptic characters are there, some Ayurvedic principles are there, but that things can be carried out by the botanists also with the support, or we can have some small interaction with the Dravya persons in that Ayurveda line. He may or she may help you to work in that. So a lot of work is remained to work in that, do research in that, and extensive testing is there. We have all the NABL labs and NABH hospitals we have for the clinical trials, because whenever we generate the data, that data will not be accepted as it is. They ask for that what are the procedures we followed, that good collection practice, agricultural practice, storage practice, what type of lab it is, that is the NABL accredited lab is there, or that NABH accredited hospital is there for the clinical treatment. Suppose that preclinical pharmacology work we are doing means where that GLP lab only they need, that GLP lab, that too especially as per the international guidelines, that what we can say that international guidelines has to be followed in that. So in that way it works. So this quality evaluation of raw materials, simply I can say in picture, organoleptic, that taste, color, odor, taste, texture we will check. Microscopy botanically we will see. Physically foreign matter, moisture or anything is there means we have to check it. And chemistry study, heavy metals, aflatoxins and other things we will do. And here marker study also we will do, and microbial contamination we have to do. Then that raw drug will be cleared for the further. If it is failed somewhere means that is a waste here. So like that we have to plan our work. Especially in the pharmacology, what that industry needs, the same thing we are doing, that the same thing I am presented here. Generate scientific evidence. Sometimes we collect the data from the traditional healers, but we will not collect the data how he identified the disease. When the patient reaches to him, is he carries any that information, that test reports with him, or if he is using any of that allopathic medicines to prove that he is having the particular disease of that. That preliminary some data we need. Then that doctor, that healer will treat it. After the treatment, we are not going for the further study. We have to check it that really cured or not. That when we have to test it, that data we need that. So like that here the scientific evidence is very, very important to promote any formulation in the society. So here that generate scientific evidence on a safety, that the drug should be that should be a safe one, especially for the Ayurvedic herbominerals formulations also there. So where we are taking utmost care. Biological screening of Ayurvedic drugs for efficacy and biological activity, and this generation of evidence in the safety of Ayurveda medicines. In that we worked many things, 160 drugs we worked, and 54 single drugs. It means a single plant material where we used, and 31 Ayurvedic formulations which are available one, and 75 coded drugs. It is already known Ayurvedic plants only, where that safety toxicity that has been studied. So this is a safety assessment, one brief it is given. Safety assessment, how we will do. Observation is there, what we say, epidemiological studies data we will collect, and preclinical data we will collect that data, and finally that will goes to the clinical. This two data, this observation and preclinical data is not available means we cannot take the clinical trials. The same thing how we will do the same thing about that clinical trials, how it will follow the different steps. Finally, we will get end product is that therapeutic, that is a medicine. Like that, some clinical research contribution is there. The same thing, that evidence, that new drug development is the main our motto. Then intellectual property rights, this is a must. We have to follow this IPR guidelines. Regular updating should be there. It is there in our all the centers. We will update to all the people, and trainings and other things will be provided to the people. And sometime we will add some of the Ayurveda universities, colleges, people to add in this. Suppose any statistical support they may need also that time, that also provided. So many things are there. And institute ethical committees it is having. Ethical committee clearance is a must for the any clinical trials. This ethical committee works according to the international guidelines, what it is there in this allopathy medicines, like that only. The same, all the guidelines has to be followed. That should not harm to anything, and no deviation in this. This is a COVID-19 simple picture I have presented here, that 46 prophylactic studies has been carried out. Before, this is a prevention thing, and 49 for the treatment, 17 for the observational studies, and some preclinical studies about 24, and survey conducted 14. At the end, we got that ISO 64 after all these thing. So some any new thing has to come out, means behind that a lot of work is there, that all that will contribute something a good thing for the society, like that. So some of the guidelines things I have presented here, that GCP guidelines from the ASU drugs, and ICMR ethical guidelines are there, GCP guidelines for the CDSCO, Ministry of Health and Family Welfare, WHO guidelines are there. All are available freely on the net. That has to be followed as per the need. And apart from that, so many other things are there, that NRDC, our National Research Development Corporation is there. This is helping all the research labs to reach that our product to the industries. And I'm at the near to the end. I wanted to tell you some of the things. You can get the benefit of our councils and our institutes. Some of the certificate courses are there for the students, and some postdoctoral fellowships we are giving to the students of related to medicinal plant research. I can say that it may be botany, microbiology, Ayurveda, related to Ayurveda. Here some microbiologists can apply for that, and botanists can apply for that, and some of the people are there even in Karnataka also. Some of the students are getting the fellowship in the botanists. You can apply that. Ayush PhD fellowships are there, studentship programs are there, PG star we are giving, and some trainings we are giving. Apart from that, I wanted to tell you one thing. Especially, it is very important for the universities. Uh, some of the lab facilities we may not get at the time of our work. So for that, our council started a initiative to help the students. If any work they wanted to carry out in our research institutes, we will allow the students, but that student has to come through the guide only, through the university professor only. If they directly approaching, means it will not be accepted. So some of the things we are doing, I can say that these are the works that student can come and do. Some minimum charges is there. If he pays, means that is enough. In that also some discounts also we are giving to the students. Especially pharmacogenetics is there, chemistry is there, preclinical trials are there, and pharmacology work, if anyone wanted to do with us, or they wanted to take help from our institutes, they can take. So this is our CCRS website. A lot of information is there. If anyone need, they can use and take the help. And even our scientists are there in the all over the India, in different states and different institutes. They can contact them and they can help. Here all are freely available to interact with you. No charge, nothing in that, but the students will be charged for their lab use and other thing. Otherwise, nothing is there. With this, I will close my talk. I think I'm in time, on time, I think so.
Okay, participants, because Dr. Shiddamallaya is associated with funding agency, that is Department of Ayush. There's a lot of fund actually, so you can approach him, and you can, if you have any doubts, you can clarify with regard to that, how to apply and what are the things to be followed, all those things.